My research is investigating Salmonella bacteria. Salmonella is a bacteria that causes diarrhea and systemic infections and is responsible for 155,000 deaths annually through diarrhea and 388,000 deaths through infections in Africa alone. So in Malawi, based in Blantyre, and at the Central Hospital, the Queen Elizabeth Central Hospital, we observed a new group of Salmonella pathogens that emerged, and they were infecting more patients. But this had similar characteristics with the previous pathogens that we observed, and so we were curious to know what exactly has this new group acquired that is allowing it to be more infecting more patients and killing more patients than the old one. So basically saving lives, trying to find answers to sickness and disease in Malawi. So my research is very important, investigating Salmonella, because of two reasons. One is because we are going to understand this new pathogen that has emerged in Blantyre, but also we are developing a laboratory technique that will be used to quickly identify new strains that emerge within the Blantyre setting and Malawi in general, that then will have results quickly, and it will inform doctors on how to treat patients, but also inform communities on how they can control transmission of these pathogens. So in this work, we're trying to understand the genetics of this new group of strains, which we have so far determined. We noted that this new group of strains is less mobile in the human gut, and therefore evades the immune system, doesn't initiate diarrhea symptoms, and then it invades the patient. And in there we have also understood the type of genes that it uses to be more virulent in patients. But also we have computationally designed the laboratory technique that will be used to be able to detect these organisms on the real hospital setting. We have done this computationally, but now we are testing the technique in the lab environment. So those are two major outcomes of this work. The AESA-RISE funding has really made me as a scientist. I don't know what I could have been without AESA-RISE, because the skills that I've gained in this funding has connected me to other bigger projects. I'm currently collaborating with the rotavirus research, where we're investigating rotavirus in Southern Africa. Recently, we have started to use the transcriptomic skill that I've gained through this work to investigate the single-cell biology in infants. And so it has really linked me to several bigger grants. We recently acquired some servers for data analysis through collaborations, but also we acquired a huge single-cell sequencing machine because of the skills that I acquired in this grant that allowed me to collaborate with others. But also I've trained master's students. I graduated 3 students in the previous cohort, and now I've got about 3 more master students that I'm working with. So that allows me to transfer skills to the new generation, which will be able to carry on with the skills. My next steps now is to diversify, using these skills that I've gained in this, is to diversify my areas of research, applying the skills here, apply for more grants, train more masters, PhD students, but also postdocs, and build a research team that is strong that can be considered to be a center of excellence in computational biology, microbiology, as well as the molecular biology, investigating different issues. So it's to build a research group and transfer skills to the younger generations.